Hello, I'm Mandy Sander and I'm a PhD student at the University of Duisburg-Essen and will now briefly present you my project. eDNA-based bioassessment for benthic invertebrates typically uses the mitochondrial cytochrome oxidase 1 gene region for metabarcoding, but due to the codon degeneracy of the CO1 region, the target reads are watered down, making the majority of reads non-target. Therefore, when using universal CO1 primers like BF2 and BR2 that were used in our study, the percentage of target reads is less than 10%. We therefore designed a target-specific primer. To do so, we identified the most abundant non-target OTUs generated with the universal primer pair BF2 and BR2 and designed a specific reverse primer named EPTDr2n. The primer region shows distinct bases, especially for its name-giving orders EPTD, but also for other benthic invertebrate orders in comparison to the non-target taxa here highlighted in green. The new primer is located within the BF2 and BR2 fragment and uses the freshwater chew as a forward primer. For the DNA amplification with the new primer, a two-step PCR with unique tags was conducted. Subsequent steps included sequencing and bioinformatic analysis. When comparing the read abundance of target and non-target taxa with other, more universal primers, the new primer shows a much higher percentage of target reads. In this case, up to 99% are arthropod reads. Although some invertebrate taxa are underrepresented due to the difficulty in finding suitable primer regions separating target from non-target groups, the results show that with the new primer the non-target amplification bias is significantly reduced. Additionally, the primer shows an increased resolution for most insect taxa and especially for chironomids, but also for annelids. Thank you for your attention, and if you have questions left, for example regarding the in silico evaluation and the performance of the new primer for the different taxa, or are interested in more results, you can look up the paper shown here or ask me questions at the conference.